Science Friday.
Here are some for E as well. The one on the top here is one of the ways that you can define E.
What makes a field more likely to have an unbalanced gender distribution?
So this can be, like, in the case of this paper, gyrification or brain folds.
A neuroscientist started to look at the brain
computers,
as
like the
more as an
macro
interconnected
I'm going to ask
very
an incredibly negative
area.
consequence.
Okay, no one, great. Don't worry, this isn't a quest project. How many of you guys have had really amazing mentors? Yeah, quite a few. Cool. So that's what I'm going to talk about for a second, and then I'll jump into my presentation. This was actually a continuation of research that I did at a summer internship in Minnesota a couple summers ago. I was a rising sophomore, basically had no lab experience, joined a lab, and they gave me stuff to do. They trusted me. It's kind of crazy. And then during the school year, I basically reached out to them and I was like, hey, I want to continue the research. And I didn't, I don't know, cancer was interesting to me. It wasn't an area that I'd really thought about before, and I don't have any specific reason why I specifically chose to do cancer other than the fact that that's just what was available to me. But the people that I met during my internship, who are on my acknowledgement slide, they were the people that actually motivated me to keep going. And so Blake, who was my mentor, basically was kind enough to send me all the reagents and all the cells that I needed to do research. Daniel, who is one of my close friends now from that summer, basically helped me throughout the entire process. He basically helped me put together a presentation and a poster to present at conferences. So a lot of this research was made possible because of them. This entire process has kind of taught me how important mentorship is and how important it's been to me. And basically, the reason why I've continued to do research and why I want to continue to do it in college is because I've been able to form these relationships and because I want to continue doing that. So without further ado, I'll get started. My presentation, as you'll see, is about sensitizing non-small cell lung cancers to chemotherapy, basically. I'll talk a little bit about why I'm specifically focusing on non-small cell lung cancer. I'll talk about the drugs that I'm using, how I actually treated the cells, and then I'll talk about some of the results that I saw and what I would like to do next. A little bit of background. So there are two-ish types of lung cancers. The first one is non-small cell lung cancer, which accounts for about 80 to 85% of lung cancers. There's a little over 180,000 cases estimated for 2020, which is a lot. It's pretty severe, but the 5-year survival rate is around 24%, which is pretty good for a cancer. Long story short, it's basically more prevalent but not as severe as small cell lung cancer, which accounts for around 10 to 15% of lung cancers. Far fewer cases, but 70% of the patients who are diagnosed with small cell lung cancer already have a metastatic cancer before they've been treated. So something's already happening. They already have been diagnosed with cancer. It's spread, and then they're diagnosed with small cell lung cancer. The 5-year survival rate is a lot smaller, so it's a lot more severe, but still it's not as common. The other thing is that most small cell lung cancer cases are in people who smoke, and so there's already there's more causes, and there's just a lot more that happens before a patient is actually diagnosed with small cell lung cancer. Mesothelioma isn't exactly a lung cancer. There's a little bit of debate about whether it is or isn't, but it essentially affects the lining of the lungs, or the pleura. I won't go too deep into that. The lab that I was working at mainly focused on non-small cell lung cancer and mesothelioma. But for my research, I'm focusing on non-small cell lung cancer, basically just because of how common it is. There's a little bit more research being done about it, just because of how prevalent it is, and So I'll jump into talking about the drugs that I used. The idea is that I'm using 2 drugs, and I'm treating cancer cells with both of them. I'll get to that later, but I'll talk about their mechanisms first. So Topoisomerase II is essentially a mechanism that binds to tangled DNA. It cuts the DNA so the DNA can untangle itself, and then it helps rejoin the DNA. And keep in mind that this could be healthy DNA, or this could be cancerous DNA. It has no way of really distinguishing that. But for this study, we're looking at cancerous DNA. And so etoposide, which is the drug that I was focusing on, it essentially forms a complex with topoisomerase and the DNA strand. And it basically forms an irreversible bond. And this damages the DNA, so the cell eventually dies because it recognizes that something's wrong. And keep in mind, these are cancer cells. A little bit of background on the drug, though. It's already been used as a chemotherapy drug for quite a few years at this point. It was discovered in the 60s, and then in the 80s it was approved by the FDA. That's not super important, but just in case you guys like history. This is the newer drug that I was using. It's basically been used only in preclinical studies, specifically alone in non-small cell lung cancer cells. I guess I'll use a metaphor that I thought of to kind of describe this drug. Okay, so imagine there's a car factory and they're basically producing defective cars, and they're basically producing defective cars, nothing's happening about it, and finally one of the workers in the factory is like, hey, hold on, there's something going wrong. The worker, instead of telling the big boss, basically goes and breaks some of the machinery so the cars can't be produced anymore. This is essentially what 4AGI1 does. So we're specifically talking about translation of weak mRNA. Weak mRNA basically contains genetic code that'll create DNA that's cancerous. So it has signals that'll basically tell the cell to proliferate or like grow very rapidly and divide rapidly. It'll also tell the cell not to basically die. So it has anti-apoptosis factors. And what 4AGI1 does is that it forms a complex in between eIF4E and eIF4G, which form a complex that'll help translate this weak mRNA. And so 4AGI1 essentially inhibits that translation. So after treatment with 4AGI1, the ribosome can't really translate weak mRNA. At this point, does anybody have questions before I move on? Okay, any parts that are confusing? Okay. Okay. The question that I was trying to answer is, actually, one thing that I forgot. Etoposide is currently used, but it's super toxic to healthy cells. And so if you have a really aggressive cancer, you kind of have to use a lot of it, but it can, like, you see things like patients getting really, really sick. You see things like hair falling out. It's basically just super toxic, and so physicians try not to prescribe it in high quantities. And so the question that we're asking is, could you use etoposide and 4AGI1 in small quantities, but still have a greater effect than using either drug alone in the same quantity? So that's the question that we're asking. Basically what I did was I used 2 non-small cell lung cancer cell lines for H460 and A459. I used 4 concentrations of etoposide and 6 of 4AGI1. I'll show you guys like a little diagram on the next page, kind of laying out like my experiment. But I essentially found these concentrations of each drug basically by using dose dependency curves, which basically tests a range of concentrations and tracks how much cell death occurs. We kind of tried to find the ideal concentration. We kind of looked at like the range from like 20 to 40% cell death. And basically this is because like we don't want too much cell death. We also don't want like no cell death. We kind of want something like in a sweet spot. So this is what my assay looks like. This is essentially a 96-well plate. So in each one of these cells, there's its own like little reaction happening. So each is basically considered an experiment. In the 4 quadrants, we have the different concentrations of etoposide. And then in rows 1 through 6, and then again repeated from 7 to 12, we have the concentrations of 4AGI1. So each cell basically has 2 repeats. So we have like some, I don't know, basically just like trying to reduce error. We take the average of all of these whenever I was doing like the data analysis, just so that like if we had like an outlier, like we would account for that. Any questions? Okay. Cool. So this is the H460 cell ability after the combination treatment. I should look at my notes. Okay, so the red, blue, and green lines represent the combination treatment with etoposide and 4AGI1. And then along the x-axis is the concentration of yeah, the concentration of 4-AGI1, and then each of those lines represent a concentration of etoposide. And so basically the takeaway is that there's a significant decrease in cell viability as you increase the concentration of both drugs. Similar for A549, there's one thing that I don't really understand about this graph is why cell viability actually increases for 0 micromolars of etoposide, but for a little bit of 4-AGI1. I'm not really sure why that was. That could have just been error. But it turns out it doesn't actually affect the data that much. These graphs are basically taken from those other two graphs. Essentially, it shows a normalized cell viability in both cell lines for the combined treatments. And it essentially shows that for the combined treatments, cell viability was significantly lower than if you used either drug alone or if you used no drug, which is kind of what we expected to see. The white bars are the cells alone with no drugs. The red bar is only 4-AGI1, and then the blue bar is etoposide, and then the green bar is both drugs in a combination treatment. Takeaway here is that combined treatment was a lot more effective. We also used a student's t-test with the with our value. We used a student's t-test to determine whether our data was significant. It was our p-value, our threshold was 0.05, and we got a p-value of 0.02, which is pretty good. I think in conclusion, a couple things. We can conclude that the combination treatment led to significantly lower cell viability. We also know that this data was statistically significant, which I mentioned already. And we can conclude that patient outcomes could increase when treatment includes a topoisomerase II inhibitor and 4-AGI1. The reason why I say could is because we didn't actually test anything in vivo or in actual mice or things like that. It's just in cells, so we can't actually track adverse effects or how toxic the drug is to healthy cells. In the future, it'd be cool to actually test this in mice, and I don't know, that would be a while from now, but it would be cool. But to do that, we'd also have to test it in far more cell lines. Unfortunately, COVID started right when I was doing this research, so I couldn't actually do more cell lines. And then I would also like to expand it to other cancers. I'd started working on breast cancer, but again, COVID happened, so I couldn't really access the lab. Any questions for now? Yeah?
So how do you know whether your results came from having some combination of the two being more helpful versus just the more chemo, the better? So I saw that you had 12.5 units of one and 10 of another, and then together there were 22.5. Did you ever compare it to just a lot of one?
So that's kind of what this graph is showing. So you could have 50 micromolars of etoposide and none of 4-AGI1, which is over here. And yes, the cell viability is a lot lower, but also you don't really want to use either too much of either drug just because they're super toxic. And so they're going to damage healthy cells.
So they're less toxic if you use a little bit of each?
Yeah, that's essentially what we're trying to test. Yeah.
What are the roadblocks, if any, to administering both drugs at the same time as opposed to just one?
Say it again?
What are the roadblocks, if any, as administering both drugs at the same time? Is it harder to produce? Is it
in a clinical setting or in?
Clinical setting, if you could explain that.
I'm not actually sure. None of the work is this is all preclinical. I think it would be interesting to look at this in mice, and it would be actually interesting to see if you treat what? Oh, repeat the question. The question was what roadblocks would be what could be present if you were to treat or use both drugs in the clinical setting. Short answer, I don't really know. I haven't tested this in mice or anything. I feel it would be fairly easy to administer two drugs at the same time because their mechanisms don't really interact with each other. So it's not like you have to treat one or use one drug before and then treat with the other. Sorry, this is a little bit of a tangent, but the drug that I was working with over the summer actually had interacting mechanisms, and so you had to treat one with one drug. You had to use one drug before, and then only then would the second one work. Questions? Okay. Cool. Yeah, I just wanted to say thank you for listening. These are all my mentors. Dr. Kratzky was our PI, and Blake was the one who was really kind and sent me all of the reagents and stuff. yeah, so thank you.